My name is Kelly Chaves, and I am a postdoctoral researcher at UCSF, University of California, San Francisco. And I work in the Klein lab on creating a 3D microenvironment using biomaterials to house dental stem cells. I always grew up with my mom telling me, go to school, go be a doctor. And so, I went through high school, took, biology, chemistry classes, did fine in them, and, went to college and majored in biology. And I actually got a work-study job in a laboratory. They let me take samples and do protein analysis on them. And so basically they were a marine biology lab. And so they would go to the Gulf of Mexico every summer and hang out on the beach and collect all these tissues, and they would bring them back, and then I would, I'd analyze them, and I just was like, this is so cool. Like, scientists get to go hang out in Mexico on the beach and do all this stuff, and then they came into the lab, and everybody was just so cheerful, and it was just a really fun place to work. And so that's what really got me interested. I was like, oh, I kind of like this. This is kind of fun, So what I always tell people is that we're trying to grow teeth. And the way we're doing that is taking a cell line. These cells that were derived or came from what's called the dental epithelium. It's these cells that start out as stem cells, and then they, as they mature, or differentiate, they become these specialized cells that produce enamel. And so what we're trying to study is the point at which they are stem cells and the environment that they are housed in, to be able to recreate that in a dish. So we're using biomaterials and trying to create different shapes, or it's called microtopography. And so by creating these different shapes on a very small scale, like a micro and nano scale, we can try to trick the cells as if they were still in the body, and to be able to just house them in a dish instead. And then to study the cells, I do different types of staining on them, and then look at them under the microscope to see if they are producing the right markers that we know those cells should produce. So if we were able to study just the process of enamel formation and the process of these stem cells and keeping them maintained, in the future we could help people who, for whatever reason, have tooth defects at birth. We could help people who have injured or lost their teeth, as well as the elderly who have lost their teeth. It's a wide range of people we could help with this technology. The excitement of figuring stuff out, you're basically tinkering in the lab, trying to solve problems, and when it clicks and when something works, it's a really good feeling. It's yes, I figured this out, and then you just kind of move to the next thing and the next thing, and it's a fun job. Yeah.